Hey gang, so here's a tutorial on the vegan package. And I'm just sharing my screen here in RStudio. So we have talked about vegan a bit during this semester, and I just wanted to spend some time on this video showing you some of the main things that you can do with vegan. There's a lot more out there to this package than what I'll be able to cover today. This is, I think, a good introduction. So you'll need to install and then load the library for vegan if you haven't done that already. There's some dependency packages that'll be loaded along with it. And then I found on GitHub a package called ggvegan that's helpful for creating plots from some of the multivariate ordinations that I'll show you today. As I think we talked about early in the semester, sometimes packages before they're completely stable or completely developed and before they go onto the CRAN, you can download them from GitHub. And so this line 3 here is just loading the library of developmental tools or dev tools. So you'll need to install that one and then load it. And this next line of code here is what you would run to install the ggvegan from GitHub. So notice it's the dev tools, a ;: install underscore GitHub is the function, and then there's just a file path there. So I have already run that, and it will download the package onto your computer. So then after you run that, you can just run library ggvegan as you would any other library. And notice that it loaded there. So within vegan, there's some data sets that come with the package that are useful for exploring some of its functionality. We'll focus on just one of those data sets for this tutorial. This is a data set that's related to mite communities. So if you use the function data mite, what this will do when you run that is create a data frame with 70 observations and 35 variables. And if you look at that data frame, these are species identifications or taxonomic groups across the top, and then abundances going down for 70 locations where these were sampled. There's another data set, mite.env, that has environmental data. If you run data mite.env, you'll see that there is a data frame that has 70 observations of 5 variables. If you click on that, some background on these environmental variables. So this is shrub density. These mite communities are coming out of soil cores. So this is substrate density. This is water content of the substrate. This is the kind of categorical type of substrate that those cores came from. This is shrubs, so where there are few, many, or no shrubs around. And then these are topological features. Was it a blanket topological feature, or was it a hammock? And of course, you can use, if you go down to the bottom here and just do $MITE and hit enter in the help window down here, notice that there is a description of this data set. So notice it comes within, it's in the package vegan, and it tells you a little bit about each of the data sets. The last one that I'll cover today is this mite.pcnm. This is the principal components of neighborhood matrices. This is a spatial autocorrelation function that you can create in R to think about how sites are spatially related. So the PCNM is an ordination method in itself, and what it creates is a list of vectors. So I just loaded the mite.pcnm. If I click on that, there's vectors here. It goes all the way up to 22. And each of these represents spatial autocorrelation. The first vector explains the most. So you can think of this as broad scale spatial autocorrelation. And then as you start to go further into these dimensions, you're moving into finer and finer spatial resolution, and eventually at some point, just interpreting statistical noise. And where you draw the line on using those PCNMs is actually an area that has received a lot of research attention. For today, we'll just focus on these first three, kind of the broad spatial scale relationships among these 70 sites. So those are the data that we'll work with, and we'll just go through a few of the ordination methodologies that you can use in vegan. Probably the most commonly encountered is principal component analysis. This is an unconstrained or indirect ordination method. That just means that you have a single data matrix that you're creating linear combinations of the data to come up with a reduction in the number of variables. So this is a variable reduction approach. And to run a PCA in vegan, we're going to create on line 16 here an object called PCA. But we're going to use RDA as the function. This stands for redundancy analysis, and that's a form of constrained ordination or direct ordination where you have two matrices, usually a community matrix and then a matrix of environmental data, and you're thinking about how much variation in the community matrix can we explain with the given environmental data. But if you only give one matrix, what vegan does automatically with the RDA function is it runs a PCA, an unconstrained or indirect ordination method. So we're going to use the RDA function. And then I have might.env, and then I've just selected, if you remember this indexing, I've just selected the first two columns there. So if we run this, we created this object that's called PCA that should only be plotting the first two continuous variables from this might.env data set. So if I click on that, remember this is substrate density and water content. So we can look at the summary of that PCA. And what it does is it creates, as I said, a linear combination of the original data. So let's go all the way back up to the top here. We're going to talk about partitioning of variance today. When we think about inertia, inertia is variance. So how much variance can we explain with environmental data? That's this idea of constrained ordination. Because we're doing unconstrained ordination here, notice that all of the inertia or all of the variance that's there remains unconstrained. And so the idea here is that you create a principal component or a linear combination of the data that you're modeling, and most of that variation should be explained in the first axis, and then the most amount of variation is explained by the next axis, and so on and so forth. And you have the same number of axes as you do environmental data that you put in there. So we only put in two variables, and we have PC1 and we have PC2. And so these are eigenvectors, and each of those has an eigenvalue or some amount of inertia or variance associated with each of those. And what I want you to notice is this proportion explained. So of the variation that's explained, notice that 99% of that, this is 9.9 times 10 to the negative 1. So that would be 0.99. And that's a proportion. You multiply that by 100 and you get 99%. So 99% of the variation in the relationship between these two variables is explained in PC1. And then principal component 2, the proportion is less than 1%. So here's the proportion explained by each of the PCs, and then there's a running cumulative proportion which will always sum to 1 when you get to the end of all of your PCs. So you can imagine if there's a large number of environmental variables going into this, you'll have a large number of PC axes, but you should always have the most variation in the first, followed by the second, followed by the third, and so forth. So in this case, instead of thinking about water content and substrate density independently, we can create this linear combination of the two, call it PC1, and that is our new potentially predictor variable that we might want to use. Alternatively, it's just a way of thinking about how environmental variables relate to each other across the landscape. So then down here, what it gives you under species score, so it's using the word species even though we're already thinking about environmental variables. It tells us where substrate density and water content fall out along PC1, so, and then along PC2. So we usually plot these things in 2 dimensions, and I'll show you how these coordinates plot in space. And then the site score, so these are just coordinates along PC1 and PC2 of each of those 70 locations where community data were collected and where these environmental data that we're analyzing were collected. So one of the reasons that we downloaded, if we go back to the top here, this GG Vegan is because there's a function there called Autoplot that makes these really nice multivariate plots for you. So all we do is use this function Autoplot. We're going to put PCA in there because that's the name of the object that we created with the RDA function. And then we're going to do arrows equals true. And what that's going to do is add arrows or vectors onto the plot that represent these 2 environmental variables that we are thinking about. So I'm going to run that line. And we get this plot down here. I'll zoom in on this plot. And what I want you to notice is here's PC1, where the majority of our variation was explained. Remember, 99% of the variation was captured along PC1. And then here's PC2. And notice that water content has this very long vector that's going along PC1, representing that there's a strong influence of that environmental variable on that first axis. The red points, as the legend here suggests, are the sites. So the 70 locations where these 2 environmental variables were measured is what the red points represent. So now we can think about, instead of these 2 environmental variables, we can just think about PC1. We can think about sites that are positive along PC1 being sites that have a high water content, and those that are negative along PC1 is those that have a lower water content. And then our second axis that explains very little variation is a gradient in substrate density. So high densities negatively along PC2 and then low densities positive along PC2. So the length of the vector there gives you some indication of how important that environmental variable is. And so here what we have essentially done is just created these 2, we've collapsed, I should say, these 2 environmental variables into this one axis, thus the phrase or the term variable reduction. So this comes at a cost, though. It does kind of change the way that we interpret this axis. These numbers are no longer interpreted on the scale of the measurements that we use for those environmental variables. It's now this multivariate space kind of coordinate score that exists along PC1. It's one of the criticisms of principal component analysis. It's a good way of collapsing redundant variables, but it does compromise your ability to interpret what the numbers actually mean. It's all relative. So that's a principal components analysis. That's a form of unconstrained ordination where we have a single matrix and we're thinking about how can we summarize the relationships within that matrix. In ecology, most of the time though, we're thinking about communities and how do communities of organisms relate to their environment. And so this next bit of code here then is thinking about constrained ordination. So here we're still using this RDA, this redundancy analysis function, but notice now we're giving it might, which is the community matrix that we loaded earlier, and we're using that as the response variable. So remember this tilde, if it's to the left of that, it's the response variable, and if it's to the right, those are independent or predictor variables. And we're just asking with this model, essentially, how much variation in the community composition of these mites can we explain with substrate density and water content. So notice there's a plus there. We have those 2 predictive variables that we've already kind of explored a bit in unconstrained ordination. Then there's a comma, and then data equals might.env. That's just telling it where to look for what data frame should it look for these predictive variables that are listed here in the model, in that equation. So we can run that, and we can have a look at this object RDA that we just created. And so notice that this is our formula here. This should look familiar, but now we have this inertia again, where the total is broken down into constrained and unconstrained. And there's a little reminder here that inertia is variance. So constrained inertia would be the amount of variation that was explained by these environmental variables, and unconstrained would be the residuals or the amount of variation that was not explained by those environmental variables. And here's the proportion in this column here. So of the total proportion, 21% was explained by these two environmental variables, and 78% of the variation in community structure among these mites was not explained by these environmental variables. So we have these eigenvalues for the constrained axes, and that is essentially a measure of how much variation is explained by the first axis versus, which is RDA1 now, versus RDA2. And notice that most of that variation is captured in RDA1. So it's the same concept of collapsing multiple predictor variables, but we're also now thinking about multiple response variables. And we can again use this autoplot function. We'll call it on RDA, that object that we created with the RDA function this time, and then arrows equals true again, so that we have vectors for our environmental variables. And when we run that, we get a plot that looks a little bit different than what we had earlier. Notice it's now not PCA1, it's RDA1 and RDA2. Notice that water content is still the primary driver here. And all of the scrambling of information down here, this is a bunch of text representing each of those mite species that were collected from there. So notice there's this one outlier point way out here that's causing everything to be clustered down here, and perhaps we would want to remove that outlier and run it again and get a little bit better resolution in terms of how these species are sorting out along this gradient, particularly of water content. So the nice thing about an RDA is you can actually do this with more than one predictor variable matrix. You don't only have to think about the environmental variables, or you may want to think about spatial variables, so this PC and M, or you may want to take those environmental variables and break them down into the categorical variables versus the continuous variables. And I'll show you how to do both of those things. So here we're creating another object called RDA2. We're using the same function RDA. Our response here is mite community matrix. Then notice that period is there. And what that represents is that we're using all of the variables from mite.env. So if you'll remember back to earlier in the semester, we talked about the shorthand way of coding an equation. You have multiple predictor variables. If you just want to use everything in the matrix, you just put a period there. Then notice we have a plus sign. We're using the function as.matrix, and we're pulling the mite.pcnm, the first three columns from the spatial data. So remember, the first few columns in PCNM are the ones that explain kind of the broad spatial scale patterns. So we're just going to pull the first three. And we're going to run this in an RDA, and we're going to consider how much variation in community composition of mites can we explain when we bring in some of these other environmental variables, and when we consider spatial variables. So we can look at the RDA output again, and if we go up to the proportion of constrained versus unconstrained inertia, now notice about 36% of the variation is explained by these environmental variables that include space plus some of those categorical variables. But notice now we have a much larger number of RDAs, a much larger number of eigenvectors. RDA1 still explains the most variation, RDA2 the next most, RDA3 and so on, but notice this is attenuating to, at some point it's a very small amount of variation compared to the first axis. So this is kind of the double-edged sword of using these multivariate statistics is if you use RDA1 and RDA2, you're capturing most of the variation there. And in those 2 dimensions, you can think about a lot of things that are happening. How are the mite communities differing? How are environmental variables related to that? But you're essentially ignoring some of this higher level or these higher dimensions, RDA 3 through 14, for example. There may be some information in there that we're missing, or it could just be statistical noise. And this is another area that's received a lot of attention. How do we determine how many of those axes do we keep to interpret? Usually it's only the first 2, sometimes 3, depending on how quickly the inertia attenuates across those. A lot of researchers, including myself, have moved to using non-metric multidimensional scaling as a way of showing patterns in only 2 dimensions. And that's one of the things that I'll show you at the end of this tutorial. So we can plot this. If we go back to Autoplot RDA2, we'll say arrows equals true. And notice now that our figure is even more complicated because we're thinking about still the major contributor here, water content, but we also have shrub level low. We have vector 1 and vector 2 in our spatial data, and we have shrub density. And so the points are separating multivariate space. And we can think about these linear combinations of multiple environmental variables and how that relates to linear combinations of community structure. So here, RDA1, a positive score would mean we're talking about sites that have high water content and high shrub density. And if it's negative along, notice the 0 is right here. So if it's negative along RDA1, we're thinking about sites that have low water content and low shrub density, but may have high correlation on the first or the third vector of the PCNM. Our second RDA here is a gradient between sites that have low shrub density. So this vector is going negative along RDA2 versus those that are clustered together along the second axis of the PCNM. So there may be some spatial autocorrelation happening along that second RDA. But again, water content is pretty important for community structure along the first RDA. And one of the things that's useful here is thinking about, well, how can we partition the variation between these spatial measurements and these environmental variables? Is there some sort of relative contribution of space versus physical habitat? And this is something that we think about quite a bit in meta-community ecology. And so you can do this in vegan using this var_part function. So we're going to create this object called v.part. We're going to use this function var_part. We're going to use the mite community data, but then we're also going to use mite environmental data, the first 2 columns. And then we're going to use mite PCNM, the first 3 columns. And so we have our response matrix, and then we have 2 matrices of predictor variables that are separated here by a comma. And so when we run that, we create this object that's called vpart. So if I go v.part and hit enter, we can have a look at what this looks like. So here's our call to our equation. It tells us that there are 2 explanatory variables, or tables, I should say, x1, which is our environmental data, x2, which is our spatial data, the number of explanatory tables, something about the total variance that's there, and the number of observations is still 70. We can think about what proportion of the variation is explained by each of these. And that's what this individual fractions, this lower table is showing. So when we think about the pure effects of environment while we control for space, that's what this x1 pipe delimiter x2 represents. So the pure spatial effect is, environmental effect is about 18%. So we can explain 18% of the variation mite community with just environmental variables. With just spatial data, it's only about 2%. But then there's an interaction between those 2, and that's about 1% here. And sometimes it's easier to think about this as in a Venn diagram. So I'm just going go back and run the next line of code, which is just plot, and then this object that we created, vpart. And what we end up with is this Venn diagram. If I zoom in on that, what this is showing is the amount of variation that's explained by these two tables of environmental variables. So the box around the outside represents all of the variation. Variation that's outside of those circles is residuals or unexplained variation. So about 80% of the variation we were not able to explain. X1 was our environmental data. So the pure effect of environment is about 18%. The pure effect of space is only about 1%. And then notice there's this overlapping interaction. So there may be some spatial autocorrelation that influences how environmental variables are distributed across the landscape, which then has implications for the mite communities. So there's actually an interaction between those two that's captured in this middle part of this Venn diagram. So then we can say, based on this analysis, that the local environment is much more important than spatial structure for regulating these mite communities. The nice thing about variation partitioning is you can do more than two. You can go all the way up to 4 tables of predictor variables. So here in this object v.part2, we're using the varpart function again. We're still using mite as our response, but now we're using the 2 continuous variables, substrate density and water content. Then we're using our categorical variables, the substrate class, the shrub level, and the topography. And then we're using our environmental data. So that's 3 tables that we're now using. Notice here that we've got this transfo equals help. This is for transforming the community data. So the issue with using raw count data is that sometimes very abundant taxa or very rare taxa can have a large influence on your ordination. And this Hellinger transformation is a way of downweighting the effect of really abundant taxa on your ordination. So there's other transformations that are out there, like log transformation, for example. If you do question mark varpart, there's a explanation of all of the transformations that are out there. But in ecology, it's pretty common when you have abundance data to use this Hellinger transformation. So we'll run that and then just have a look at the plot. And what I want you to notice now, and I'll zoom in on this, is that we have 3 circles in our Venn diagram. So the variation explained by the continuous environmental variables was about 7%. And when we broke those out into categorical environmental variables, that was about 8%. And then when we used space, it was about 3%. But the pairwise interaction, so between continuous environmental variables and spatial variables, about 5% of the variation could be explained. Same between the categorical environmental variables and spatial variables, so forth, with all of your 2-way interactions. And then there's the 3-way interaction. So where continuous habitat variables, categorical habitat variables, and spatial variables all jointly explain some variation that's 17%. That was by far the largest amount within this. And all of that adds up to about, let's see, 47%, and 53% is left over as residual or unexplained variation. So variation partitioning, this is a pretty common way of thinking about relative contributions of different classes of environmental or spatial or predictor variables, that is to say, on influence on community data. So the last thing that I'll cover then is this idea of non-metric multidimensional scaling. So I showed you principal components analysis. That's where we're thinking about only a single matrix, and I showed you one with just 2 environmental variables. You can do a PCA on Hellinger transformed community data and get something that looks very similar, but instead of environmental variables in the plot, it's all of the assemblages of mites in this case. Or you can use non-metric multidimensional scaling. And so the kind of advent of this came about from this issue of trying to show variation in as few dimensions as possible without this problem of all these higher dimensions, PC3 through, you know, 20 if you're dealing with 20 species or more. There's 35 mites taxa here, so there'd be up to 35 axes there. The thing that non-metric multidimensional scaling does is it starts with a scatter that is based on a principal component analysis, but then it goes through these iterations of moving those points a little bit at a time, and then measuring how well those points are represented in two-dimensional space. So we're using this iterative process to come up with a way of showing all of this variation in only 2 dimensions. So we're not omitting these higher dimension axes or eigenvectors. We're only specifying that we want to work in 2 dimensions. So this is non-metric multidimensional scaling. And it's based on distance measures. So Bray-Curtis distance is one of the more commonly used one and the one that we'll use today. In community ecology, we can think about Bray and Curtis, this paper that came out in the 50s where they were thinking of how do we quantify differences in assemblage composition between 2 points. And so the community here, how does it compare to the community here? And then we may want to do that at points across an entire landscape. And so the Bray-Curtis distance metric does that. So a Bray-Curtis distance of 0 would mean that the community at point A has the exact same species and abundances as a community at point B. As that distance increases from 0 towards 1, we're having either turnover or loss of species. So this is the idea of beta diversity. You can have either new species that come about that replace others, or you just lose some species. So we could have 5 species at this location and 1 at this one, and if that's the same species that it occurs at both, then this community is totally and perfectly nested within this one, because this species exists at both. But otherwise, there's a pretty clear difference there. There's 5 here and there's only 1 here. But it gets a little more complex when we might have 5 species at both of those locations, but 2 of those are not the same. So that's the idea of turnover. This species goes away and this species replaces it here. And so there's a lot of research going into beta diversity and decomposing it into these nested and turnover components, but those come together to create a Bray-Curtis distance that's increases towards 1 as we see complete turnover of the community. Okay. So that's what we're trying to ordinate in space with this MetaNDS. So the function here is MetaNDS. There are a lot of defaults that are assumed here. So if you do question mark and type in MetaNDS, and it comes up in the help section here. This is from the vegan package. We're assuming a few things here. So distance, for example, is Bray. This is the Bray-Curtis distance. K equals 2. That's the number of dimensions that we want to think about. They're kind of assuming that you want to think about 2. This is the number of iterations that we're going to try to show those things in 2 dimensions. And there's a lot of other kind of technical stuff that we won't go into here. But to run it, notice that you really only need to call MetaNDS on a community matrix. So I'm going to hit run on that. And what I want you to notice, it went really fast, but it started out. So first of all, notice it does a square root transformation and Wisconsin double standardization. It's automatically transforming the data. And so if you go back over here to some of these defaults, notice this auto transform equals true. So the developers of vegan are trying to help us out here. They don't want us to run just raw abundance data because they recognize there's this issue of abundant taxa having an undue influence on the ordination. So there's an automatic transformation that happens there. If you load in data that have already been transformed, you would want to do auto transform equals false, okay? So it's telling us right there the first thing that it did was transform the data. It starts with a PCA, and it considers this two-dimensional stress. So how much does it have to contort all of those points that would be shown in 3 dimensions or 4 dimensions or 5 dimensions to show it in 2 dimensions? And kind of rule of thumb here, if your two-dimensional stress is less than 0.2, then it's okay to interpret the graphic. And as that number declines past 0.5 and anything below a stress value of 0.1 is ideal. We can have a lot of confidence that our two-dimensional representation of the data is as good as thinking about 3 dimensions. If your stress is higher than that, then you might have to change k equals 2 to k equals 3. So you'll actually be thinking in 3 dimensions. And so we can monitor this stress, and what I want you to notice is that it's iteratively changing the position of those points with the idea of minimizing that stress. And this try equals 20 is the number of iterations. And notice it says no convergence here. So it did not find a solution that minimized the stress. So we might want to increase this, this try max beyond 20 to see if we can reduce the stress any further than that. And if you can't reduce the stress and if you can't reach convergence, then essentially what you have to do is up your K to think about a higher dimension. So we'll just go with the 2 dimensions here. This stress value of 0.15 is right between 0.1 and 0.2, this kind of rule of thumb, or an area where we think we can interpret the two-dimensional data. Let me show you what it looks like using this autoplot function again. Notice here I've added geom equals quotes around text. So instead of points in the plot, it's going to be text. And then I turn the legend off because we don't need a legend in this case. So here's what that plot looks like. If we zoom in on this, what I want you to notice is we still have these latent axes. There's NMDS1 and there's NMDS2. But we don't have an NMDS 3 or 4 or 5, so we're not having to worry with this issue that there may be higher levels, higher dimensions that we're ignoring by thinking about just these 2. The red points are the numbers, actually, are sites numbered 1 through 70, so that gives us some indication of the identity of the community sample. And then the kind of greenish color are the taxa. They're not all to the species level, so these are taxa that were identified. There was 35 of them, if you'll remember. And so now what we can think about is gradients in community composition. So along NMDS1, if the community is positive, so for example, if it came from site 57, 62, 67, 59, 44, 54, anything positive along this, those communities would tend to have higher abundances of these taxa that are positive along NMDS1. As we go negative, notice the zeros right here, as we go negative along NMDS1, collections from these sites would have higher abundances of these organisms, of these taxa. So we have this gradient now in community composition. And the first axis will always explain the most, and then everything that's left is captured in 2. So NMDS1 is the first one that we interpret, and then whatever is left is NMDS2. I don't know anything about mites, so I couldn't tell you what, if these taxa up here are different from these taxa down here. But in that fish paper that we were going to read together, there was some meaningful patterns there. So I don't know if anyone in this class is interested in mites, but this is where, some understanding of taxonomy is pretty critical to understanding the ecology of organisms. And I understand nothing about mite taxonomy, so I can't help you much more there. But the point is, you could replace these mites with any data set of organisms that you're interested in. I know half the class is interested in birds. Some of you may be interested in invertebrates. Some of us are interested in fishes. You can use this same methodology for community data regardless of the organisms that you're studying. Okay, so an alternative to that, that's the autoplot. And the nice thing about autoplot is it's just like ggplot. It's the same grammar of the graphics. You can go into the documentation of the GG vegan, and you can change the background, you can change the text size, you can change the text color. You have a lot of control over these plots. And that's, I think, the way of the future, at least for the next several years, is this kind of grammar of graphics is what everyone's going to be getting on board. So all that time you spent learning ggplot is going to benefit you even here in vegan when you use this GG vegan. But let's go back just for a moment here and use the very basic plots. Because what I want to show you is just one of the patterns that was obvious to me in this MITE data set. So the first thing I'm going to do is think about the MITE environment. If we go back to mite.env, notice there was this shrub. There's 3 categories there. There's few, there's many, and then there's none. So it could be that MITE communities differ at locations that have high shrubs, or I should say many shrubs, few shrubs versus 0 shrubs. And so I just want to show you how we could test that hypothesis using non-metric multidimensional scaling and some of the extensions from that methodology. Okay, so we're thinking about how does MITE community composition differ across these different classes of shrub abundance. So because there's 3 levels there, we're going to create 3 colors for our plot. We're going to do red, blue, and green. So we're just creating an object called COL. We're using the concatenate function, and then notice I put quotes around these because these are the kind of generic or standard colors that R interprets. We can do the same thing with shapes. I'm going to do shape equals, and then if you Google shapes in R, there's numerical values that go with all of these different shapes: circles, triangles, squares. And I just grabbed 18, 20, and 15, just randomly grabbing shapes that we could plot as these different groups. So what that's going to do between those two is allow us to plot, instead of having all of these names as the same text and the same color, we can create groups that differ in their shape and in their color. Then the next line is just going all the way back to very basic plotting stuff that we started with. So it's the plot function, going into the object that we created right here called NMDS and pulling out points. So remember, the $ is used to go within an object and pull out some particular portion of it. In this case, the points represent all of the XY coordinates for the sites where these numbers were plotted. We're using color is equal to, so we're using the same thing we created here, COL. And notice we're multiplying it times mite.env and the levels of shrub. Same thing with point character, PCH. This is equal to shape, which is the thing we created here, multiplied times mite.env, the levels of shrub. There's a comma there, meaning that we go on to the next line with this code. CEX was character expansion. So what this does is multiply the text by 1.2, so it's going to make it 20% larger. We can use main equal to with quotations around MITE shrub group. So that's just the main title of the plot that we're going to create. XLab is X label, so we'll call that NMDS1, and YLab is Y label, so we'll call that NMDS2. So if I click here and just hit run, we get a plot that shows NMDS1 and NMDS2, and instead of all of the sites just being numbered with the same text, what we now have are sites that have different levels of shrub abundance. And there's a really nice add-on from Vegan called OrDSpider that will actually help us to show what those groups represent. So you can use this function OrDSpider. When you've created a biplot like this, you can overlay OrDSpider. So OrDSpider is only going to work if you've already called plot on a generic plot or on an OrDplot, like we've done here. Okay, so we'll use the function OrDSpider. We're going to use it on the NMDS object that we created earlier. We're going to define the groups. That's the whole point of OrDSpider. It's going to create a spider graph where all of the members of the group are connected by a line, so it ends up looking like a spider web. And we're going to set those groups equal to mite.env with the levels of shrub. And then we can say yes or no, do we want to put a label over that in the this case we do. So I'm just going to hit run, and it's going to add to this existing plot these spider plots. If we zoom in on that, what I want you to notice now is that we have, you can see why you might not want these labels on here, because they do kind of obscure the data. But green represents collections or communities of mites that came from locations that had many shrubs. The blue represents communities of mites that came from locations that had few shrubs. And the red points represent communities of mites that came from locations that had no shrubs. And notice that there's a very obvious gradient along NMDS1 for each of those, where we go from many to none along this NMDS1. There is an ecological gradient there. We might want to test a couple things based on that gradient. So we're near the end here. If you're feeling some exhaustion from this, the first thing we might want to do is just test whether or not these groups are significantly different from each other. So does the community composition from sites that have many shrubs differ from the community composition at sites that have few shrubs versus none? To do that, we use another function in vegan called Adonis. So I'm creating an object here called test1. This is a permuted multivariate analysis of variance, sometimes called per MANOVA. In vegan, it's called Adonis. That's the function. Our response variable is the mite community data. And we're going to try and predict that using this class of shrubs, this many, few, or none. And that class of shrubs came from the data mite.env. It does this through a permutation test. So that's the permuted multivariate analysis of variance, where it's repeating the test, in this case, 990 times, and then looking for emergence of a significant signal from those permutations. Most of the time, there's 999 is once done. If it's computationally intensive, you can do a smaller number of permutations, but the vegan package in general, and Adonis in particular, is very stable and tends to run very quickly. So we can do basically 1,000. And then the method here is just the method used to create distances. So Bray-Curtis distance is what we're using again. So I'm just going to run test1, and notice it went very quickly. It created an object called test1. If I just run the name of that, we can have a look at it. And so here's our formula. It's just reminding us of that. It tells us how many permutations we did. And then it's giving us a table of summary statistics. And it's telling us about these classes of shrubs. Remember, there were 3 when we're calculating degrees of freedom, or DF. It's always n-1. So because we had 3 groups, 3 minus 1 gives you 2 degrees of freedom. There's the residual degrees of freedom, and then there's your total degrees of freedom, which is your total sample size minus 1. So I know this is not a statistics course, but degrees of freedom are essentially how much power do we have to run a statistical test, and higher degrees of freedom are always a good thing. So that's why larger sample size is usually better for the rigor of statistical testing. So we won't go into sums of squares or mean squares, but the ratio between those gives you an F statistic, or a test statistic. And usually the larger a test statistic, the more likely you're going to have a significant relationship or a significant finding. So actually the p-value over here, this probability that we're going to find an F value that is equal to or greater than 9, the probability of that happening is 0.001. And usually for significance, we use a threshold of 0.05. So the probability of getting a number larger than 9.03 is very small, meaning there's a significant difference among these groups. If that F value was much smaller, 1.1, for example, instead of 9.0, that p-value, the probability of finding a larger value, would increase. And when it increases past the level of 0.05, then we cannot conclude that there's a significant difference. The other thing that Adonis does is it gives you an R-squared value. It tells us that about 21% of the variation in mite community structure is explained by this shrub factor. The residual, or 78%, the remaining, is unexplained. And then there's your total variation. And then it gives you a breakdown of the significance codes. This is something that R does, but I usually interpret the p-value directly. So we can conclude from that there's a significant difference in mite community structure among sites with many, few, and no shrubs. There is one critical assumption that comes with running that Adonis test, or that permuted multivariate analysis of variance, and that is that your variance within each of the groups is equal. So the amount of community variation within the mini group, the amount of variation within the few group, and the amount of variation with the none group should be equal in order for this test to work appropriately. And we can actually test for that using just a couple of lines of code here. So I'm going to create this object called distances.mite. I'm going to use the veg_dist function to create pairwise distances. This is just basically creating Bray-Curtis distances between all of those 70 locations. So I'll run that, and we get this object called distances.mite, and there's 2,415 pairwise comparisons. Here's between the first two. We get a Bray-Curtis distance of 0.46, and so on. Remember, those Bray-Curtis values will range between 0, which means identical communities, towards 1, which would be complete turnover. Then we can take those distances that we just created, and we can put it into an ANOVA and a beta dispersal function. So basically thinking about, are there differences in dispersion of the data within these distance classes or within these distance values that we just created based on the shrub levels, okay? So we're nesting a couple functions there. Beta dispersal is nested within ANOVA. And so we can run that and then have a look at the output of that, which I called test 2. So this is analysis of variance table. We've got our degrees of freedom again. Some of these numbers should start to look familiar. Sums of squares. Here's our F value. Notice now that the F value, our test statistic, is 1.01. That's a very small number. The probability of getting a value as large or larger than that is 0.36. So 36% of the time, you're going to get a value that's larger than that. That's greater than 0.05, our kind of traditional threshold for significance, which means that we can conclude that the dispersion or the variance within these groups is equal. So it's actually okay to run that Adonis test that we ran up above. The thing that I'll end with then is this idea of, well, okay, so we've got these groups that are distributed across NNDS1. They're related. At least 21% of the variation is related to many, few, or none of the shrub density. But we can also compare that to some of those continuous variables that we were thinking about at the beginning. In particular, substrate density and water content. And you can test for significant correlations with continuous variables using this NVFit or NVFit function, okay? So here we're using NVFit. We're creating an object called test3. We're using the function NVFit. Our response is the NMDS that we made earlier. And then we're trying to predict community structure in that NMDS using substrate density plus water content. And those variables are housed in the data frame mite.env. So we can run that. And we get this object called test3. And if we have a look at that, now we have these two environmental predictive variables. We have their coordinates along NMDS 1 and 2. We have a test of significance, so the probability that there's a significant relationship between that, or I should say a test of a significant relationship between that environmental variable in the community structure. So 0.006 means there is a significant relationship there, and about 14% of the variation in mite community structure can be explained by substrate density measured on a continuous scale. The more interesting one, I think, is water content. Notice that the R-squared there is 70% of the variation in mite community structure is related to water content, and that is a significant level. We have evidence that there are relationships between the categorical environmental variables plus now these continuous ones. And we can show that on our same NNDS plot by just calling plot test 3, just like we did with OrDSpider. We have to have an NNDS plot that's already pulled up. We've already overlaid OrDSpider. Now we can call this plot test 3. And I click on that, notice that we get these vectors or arrows that represent, just like in the PCA when we started this lecture, that is the direction and magnitude of that environmental variable. So from all of this, we can conclude that there is variation in the community structure of mites across this landscape, across these 70 samples. It's related to shrub density classes, the none, few, many, and they're distributed across the landscape along a gradient of water content, where there's lots of water, there's no shrubs, and then as water attenuates along NNDS1, we see more shrubs moving in, and the community composition of mites changes along the way. So there's a lot more to vegan beyond this, but that was kind of an overview of some of the main functions that my research group uses vegan for, and this may be something that helps you guys either now or in the future with your own research. So I just saved this. I'll upload this vegan lecture R script that I just made onto eCampus, and then I'll load this video as well. So thanks for your attention, and I'll see you later.